Great. Thank you so much for the time, and thank you for being here. This is our second year being part of ARM as a sponsor, and we are a platform company based in the San Francisco Bay Area. We have a unique technology that enables analysis at the single-cell level of both molecular genotypes as well as protein-based phenotypes at the same time from the same cell. We have dedicated quite a lot of our resource and commitment to actually evolving this platform to apply that to specific applications so we can accelerate the comprehensive characterization of cell and gene therapies in order to work with this community and accelerate the success of this effort. So it's been a very exciting couple of years. Uh-oh, am I going the right direction? Sorry, I'm trying to, what do I do? Here it is. It's really without saying that all of us really are committed to this field because of our patients. We celebrate them as really heroes for us and for advancing the field. Left-hand side, I'm sure is not foreign to you. One of the very first pediatric patients, Emily Whitehead, who received the CAR-T treatment 10 years ago. And she is now a healthy 16-year-old cancer-free. In the last 10 years of her treatment, I believe there's about 1,000 patients who receive the same treatment. That's about 100 per year. But the patients actually who have her same abnormality is over hundreds of thousands per year. So we have ways to go to actually democratize the therapy and make that more broadly applicable. The same goes true on the right-hand side, Victoria Gray. I really always admire patients like her with her courage for sickle cell anemia. We are anticipating, I think, huge success for the field next year, hopefully with the approval of the therapy. So far, though, in the last many years, we do know patients suffering the same abnormality exceed hundreds of thousands and actually to the millions if you count worldwide. So as a field, we still have ways to go. What do we need to do to actually begin to really move the needle and move that really magnitudes and magnitudes farther to actually really truly help the patients? One of the more recent surveys that MissionBio did was really survey the drug developers. We really believe these are the partners that we need to support. What are some of our joint challenges? Many of those really actually surround characterization. It varies, it changes from how do I do, what do I characterize, to what actually it means, both for near term as well as for longer term. If we peel back the onion just a little bit to ask deeper, what do you mean you have challenges? What kind of things do you use today, and what are you not satisfied with? Really go into two broad categories. The one relates to platform, the second one relates to support. On the platform side, as creative and innovative our CRISPR and other cell editing and cell therapy and gene therapy methodologies may be, we are actually lagging behind in the ways to analyze and treat them, to characterize them. So many of us are still living with the technologies we have lived with for the last decade, the classical ones, from flow cytometry, digital PCR, to bulk NGS. Actually, the field needs more. The field actually says, I want to be able to consolidate them. Is there a dedicated method to actually look at the characterization? Can I actually use less samples? Can I actually get away from bulk, because bulk only gives me average? So those are the challenges we believe our colleagues in the developer side face on the platform. They also face similar challenges on really, do you get the right support? Especially in this economic environment, as opposed to every single one of our developers goes to do your homebrew tests, spending millions of dollars trying to come up with your own analytical methods. Why couldn't we consolidate that and actually have those support provided to you so you can use your precious capital doing the clinical trials, doing actually the patient management, trying to get them reimbursed so collectively we can move faster. That's where MissionBio comes in. We really believe we're dedicated to providing support on both fronts to you so collectively we can accelerate and move faster. On the platform side, we talked about, which I'll go into a little bit more detail, on our differentiated single-cell platform called Tapestry that can provide you with high-resolution characterization on both the genotype as well as phenotype. We actually have two different business models. We don't just provide the instrument and reagents to you, so you struggle on your own. We also have a pharma asset development service we believe are your true partners. We can leverage our internal bioinformatics as well as in-house lab experience to actually take your assay, design those, and it can either transfer that back to you or to your designated CRO or CDMO. So you can actually scale to use that for both your internal development as well as your clinical studies. We're also spending a tremendous amount of time talking to many of you, as well as our academic pioneers, so we can better understand, actually speak the same language. Cell and gene therapy is so exciting, but there's so many different parts that have to come together. So we're going to do our part to deepen our knowledge to understand your true pain point, so we can actually help to use the latest technology on the technology side to help you solve those. So we also do think about this and believe where's the touch point, right? From both the patient journey as well as the journey of the developers. We think we can actually have multiple touch points to help you. Starting the upper right on the patient is actually start with the patient. Many of our cell and gene therapies are designed to treat cancers, in particular hematological cancers. And that's where Tapestry was initially designed to do, actually to do patient stratification, to help you understand the clonal architecture of the heterogeneous cancer and what actually are the molecular bases that constitute each of those clones. And that characterization can potentially help you better structure your therapy, design that, as well as design your clinical trials to be more successful. With that level of a deep characterization can surely help you to better understand and accelerate your development program, and then use that for lot release for either autologous or allogeneic. And then in the farther future, we also do know, like patients like Victoria still does worry about what's going to happen to the cells she's received over the next decades. Are they going to continue to produce the proteins that they're supposed to? Are they potentially, because of the DNA changes into those cells, can they become cancerous? And so there is that impact of long-term monitoring. By understanding the genotype of the cells, we can actually help you to understand and distinguish between the transfused cells versus the endogenous cells, as well as cell types, and are they still continuing to marching down the journey as we expect them to. So the reason that our platform is so unique is because we have designed the Tapestry platform to be able to understand DNA by genotyping and sequencing, as well as in the same workflow, protein from the same cells. And we're really the only platform who can do the genotype plus phenotype, while all of the other single-cell platforms really have been focusing on transcriptome analysis, which is important, but it really doesn't speak to what we have actually gone to do, which is really making DNA edits and changes in the cells. So the reason we're able to do that is because we actually have a two-step Tapestry microfluidics. In the first step, where you introduce actually protease, and the protease can actually disintegrate the chromatin structure, expose the DNA, and then only in the second microfluidic step, we introduce the cell barcodes as well as multiplex DNA, multiplex PCR, so we can amplify out the areas of interest. For protein analysis, we introduce, in the first step, pre-incubation of antibody conjugated with oligonucleotides. And so we are actually translating the protein signal to an oligo signal. Therefore, we consolidate the readout on NGS, so it's actually tags on seamlessly to the NGS platform many of you are familiar with, to give you that genotype plus phenotype quantification. Let me share a little bit with you on the assays we actually have designed already, specifically for the cell and gene therapy market. It really impacts both the virus, for example, introducing a healthy copy of gene on the left-hand side, or to the right-hand side, where we're actually doing gene edits inside the cells. The type of assays that we have developed include transduction assay, including vector copy number, includes on-off target, co-edits, zygosity, as well as to be able to look at translocation as well as chromosome stability. More importantly, all of these can be in conjunction with surface protein phenotype analysis, so we actually know if those edits are happening in the cells and what the cell fate might be in actually in your manufacturing, in your development, as well as in patient bodies. We hope to do that to provide you with a series of benefits on the left-hand side, higher resolution, to be able to detect the rare events that you actually may need to care about, at least give you that data. To have a rapid optimization, because you can consolidate all of those measurements in a single workflow with a multi-omic and multi-attribute analysis. And again, I cannot analyze emphasize this more is that we do help you with bioinformatics support if that's what you choose to do, to really save your time and make your work a lot more efficient. So how does that compare then on those particular assays? Just give you some example on what is being today done more broadly, which is really the bulk assay. Here are just some examples. For bulk, for vector copy number, you can get an average of all of the cells. But with a single cell-based tapestry analysis, you can actually begin to correlate on a cell population basis that particular copy number, whether it's correlated or not with the CAR expression. That's just as an example. Transduction efficiency, you can begin to look at VCN on the per cell basis to look at the distribution of population. I'll show you a little bit more data so you can actually get a sense on exactly what the output might look like. And then we can look at the edits. Many of you are beginning to be extremely creative and looking at multiple edits, knock-in, knockouts, and do they actually have the co-edits and the zygosity of that in the same cells. And the last two really relates to what the cells may or may not do in vivo. Is that, are they stable? Do you have other events? Are the chromosomes continue to behave the way we think they should over the decades that the patients will have to live with the irreversible treatment that we're introducing to them? So I'm going to start with a couple of slides on some of the data, very exciting data, as one of the first commercial platforms to offer VCN to this market. Now, this first one is to actually just demonstrate the correlation with the bulk sequencing, bulk analysis. This one particularly is based on digital PCR. By using clonal cell lines as well as a mixture of them, so we actually have the known truth. And digital PCR numbers, quantification of copy numbers on the x-axis, and tapestry single cell analysis on the y-axis. And so you see quite straight lines. So we have very good tight correlation between, on the bulk side, between tapestry as well as digital PCR. Now the question becomes, what additional information do you get by using tapestry? That's highlighted here. So the two samples that you actually, on the bottom, you see VCN average is actually very similar. So if you use your digital PCR as a bulk assay, you would assume these two manufacturing lots or these two samples are very similar, but indeed they're not. Because if you look at the distribution of those cells with varying different copy numbers, you see it's quite drastically different. Now, we hope that with these finer detailed, higher resolution analysis will give you some clue as you track how these therapeutics behave in your preclinical or your cell lines or even patients, and how that may potentially have an impact on actually the therapeutic response. The next example, I think for those who are using CAR-T should be very familiar. This is an actual example of one of the customers who is looking to do a triple knockout of 3 different loci. And on the right-hand side, you see the single-cell data will be able to give you actually better understanding on the 27 permutations of those combinatorial of single knockout on one loci, double knockouts on one loci, all the way to actually desired number 27 permutation. So we actually give you the distribution of that as opposed to actually going in blind and not actually knowing if you do have the desired population in your optimization of your development, as well as in the patient samples. Here's an example of actually a multi-omic approach. So you see on the right-hand side, there are 2 graphs. The top one is a delineation of the 5 cell types by looking at the genotype. So you can look at the distribution of the percentage of cells that have wild type or one allele change or actually biallelic. And they are actually clustered based on their protein expression. So this is a co-analysis of the same cell population based on our phenotype protein analysis. So there are varying cell types, and so it's very important to understand actually the different cell types. Do they still maintain the type of edits as you desired, and do they continue to do so in the long-term patient care? And so this is where we believe you can offer tremendous value, is that now you can use a single assay to be able to consolidate all of those and actually analyze them based on a specific cell type or based on specific editing requirement. So if you put all of that together, even with the existing assay, we can offer tremendous value. This is a case study of one of our customers using our platform, actually combining 4 different analyses, 4 different metrics, including the CAR copy number, including the edits, including the protein panel, looking at both on-off target as well as the control to give them a full understanding and characterization of the therapeutics. We also have had a great success in transferring the assay to our CDMO partners to support the clinical trials of our partners so they can actually have a consistent methodology all the way from development to CMC, all the way to delivering on that to patients. So this is my last slide, is to highlight that for us, the most important part is to continue to support the market, support the field, and we are here truly to be your partners and to be part of ARM, as well as to continue to collaborate with our scientific advisory board. And we look forward to leveraging our innovation to combine that with your innovation so that we can have many, many, many more faces, such as Emily and Victoria. In the future, is really, they no longer make the news. We just celebrate their heroism as a past, as really the beginning of an incredible journey. So thank you so much.